What? Camera's on? What? I'm Matt Kimball. Our NERRS Science Collaborative Catalyst grant focuses on oyster reefs, a common habitat in estuaries throughout much of the reserve. Oyster reefs support a diverse and productive community of organisms. And traditionally, techniques to examine oyster reef habitat use have been largely labor-intensive and destructive, getting out there in the reef and turning things over and grabbing things. There's some novel techniques that might get around this: high-resolution acoustic imaging, eDNA metabarcoding, stable isotopes analysis, and oyster disease assays are a few. And so our catalyst is going to put all these methods side by side at oyster reefs within 4 reserves from North Carolina, South Carolina, Georgia, and Florida, and then compare similar metrics between each of these 4 reserves and reefs within the reserves to kind of see what are the best practices for examining the ecological function and use of oyster reefs, and what managers of the reserves might do to supplement data they're already gathering on oyster reef as habitat for organisms.
Because we have partners spread throughout the Southeast, we'll have 2 planned workshops during which our team will generate targeted research questions to be addressed and develop a proposal to be submitted for future funding opportunities. These traditional sampling techniques include classic reef measurements of oyster density, percent cover, rugosity, and clump height, as well as taking nectin samples with nets and traps. The high-resolution acoustic imaging sampling method uses sonars that allow us to non-destructively examine broad patterns in abundance, size, and behavior of fauna, and to evaluate habitat function and change over time from in-situ data. eDNA metabarcoding is another non-destructive sampling method that allows us to use water samples to elucidate diversity of oyster reefs by capturing, extracting, amplifying, and sequencing environmental DNA. Stable isotope analyses give us insight into trophic structures of oyster reefs from samples of dominant producers and consumers common to all sites. Oyster disease assays are completed by collecting tissue from oysters to determine prevalence and intensity of parasite loads in individual oysters. Through this project, we look forward to providing data to feed directly into management of coastal habitats and fishery resources for NERRS management and more broadly as well. Thanks for listening.